Good afternoon. I'm Dr. Brian Shapiro, a market segment manager at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series. It's entitled The Development of Standard In Vitro Models for Studying Metabolic Diseases and is presented by Dr. Aaron Cypess. Dr. Cypess is a senior investigator at the National Institutes of Health. His research group uses a combination of clinical trials and basic science to help understand how brown fat works at the levels of the cells, and then to translate the information into practical ways to treat people with obesity, diabetes, and other chronic illnesses. In this presentation, Dr. Cypess will present on the hTERT-immortalized brown and white pre-adipocyte cell lines that his laboratory, in collaboration with ATCC, have developed and characterized. Dr. Cypess will then demonstrate that these immortalized primary cells maintain the adrenergic signaling, lipolysis, and thermogenesis typical of primary adipocytes, and that these two cell lines are metabolically distinct. If you have any questions for Dr. Cypess, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. Any remaining questions, as well as the recorded webinar presentation, will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome Dr. Cypess.
Thank you, Dr. Shapiro. It's a pleasure to be here. I'm looking forward to speaking with you about the development of standard in vitro models of human white and brown adipose tissue. And the objectives here today are first to describe the functional and physiological roles of human white and brown adipose tissue, also known as WAT and BAT. We'll then appreciate the physiological responses to chronic treatment with the beta-3 adrenergic receptor agonist mirabegron. We'll then identify the types of in vitro model systems that can be used to study human adipocyte physiology, and then finally understand the derivation and then the genetic, molecular, and functional characterization of the new immortalized clonal human white and brown pre-adipocytes. We'll begin with a slide many of you have seen before, perhaps, and that's the rates of obesity and diabetes over the last nearly 30 years. And certainly we can see that the rates of obesity are increasing, and in parallel with them, the rates of diabetes are increasing. And that's because there is a pathophysiological connection between obesity and diabetes. But even with a slide we've seen so many times, many questions can come up. For example, is adipose tissue simply something that in excess causes diabetes? And our answer would be no. We need to reassess our understanding of human adipose tissue. It is polychromatic and found throughout the body with functional diversity. And shown here are some images from a review article that I wrote in the New England Journal of Medicine. And you can see here in a frontal view, the brown or beige adipocytes in brown color can be found in certain regions of the body, in the cervical, supraclavicular, and axillary regions, also along the spine and the paravertebral region, and in distinct locations within the abdomen. So in contrast to the BAT or brown adipose tissue, there's also the white adipocytes making up the white adipose tissue. And those can be split into subcutaneous and visceral. The subcutaneous here in the orange are the ones particularly under the skin. The white adipocytes and the visceral are in the epicardial, omental, and mesenteric regions, and also the perirenal depots. And there are differences in their metabolic impact. Subcutaneous white fat is supposed to be metabolically neutral or possibly even beneficial, while the visceral adipose tissue is what is associated with cardiometabolic disease. Now, it's not so simple to appreciate the distribution of adipose tissue simply looking at an individual. For example, this is a CT or a CAT scan of two different men. This one is aged 37, and the one below is aged 82. They both have the same waist circumference of 91 centimeters or 36 inches. But you can see here that the younger man has a large amount of subcutaneous adipose tissue in orange, while the older person has considerably more adipose tissue in his visceral regions. And that would suggest that even though they have the same waist circumference, the person who's 37-year-old has a much more beneficial cardiometabolic profile. Now, human white adipose tissue begins to develop in the second trimester of pregnancy. By birth, both the visceral and the subcutaneous depots are well established. Brown fat, or BAT, first arises during the late second trimester of pregnancy, and it protects newborns from cold while they develop the ability to shiver. In lean women, the entire WAT depot ranges from 20 to 30 kilograms, or 30 to 40% of total body mass. And in lean men, it is 10 to 20 kilograms, or 15 to 25% of total body mass, and therefore, by size, one of the largest organs in the body. Now, in adipocytes, the form reflects its function. So white adipocytes are responsible for energy storage, and they have very large single lipid droplets. The white adipocytes are responsible for energy storage. They also can provide some thermal insulation. They're also important for mechanical protection. And as we'll discuss more, they can be an endocrine organ. On the other side of the other are the brown adipocytes, and they're responsible for energy expenditure through their numerous mitochondria, which lead to thermogenesis, the generation of heat. And these too may be an endocrine organ. In between the white and the brown are what we call beige or bright adipocytes. And what happens is that cold and adrenergic stimulation can lead to a transition from a white-like to brown-like functions. Both the beige and the brown have the ability to generate heat, as does the white adipocytes through a different set of mechanisms that we will review next. Shown here is the classic description of the uncoupling thermogenesis. If you recall, there's the TCA cycle that generates hydrogen atoms that are then split into protons, which go into the mitochondrial intermembrane space. The electrons are carried through the electron transport chain. They eventually need oxygen to make water. There is a buildup of protons in this intermembrane space that then go through the F1, F0 ATPase and eventually lead to the production of ATP for ADP. The ATP can then be used to drive many cellular processes. Now, uncoupling protein 1, which is seen only in brown adipocytes, has the ability to allow, in response to beta adrenergic agonists, the protons to come back down their electrochemical gradient. By going through UCP1, all of these reactions, the electron transport chain, and even the TCA cycle speed up. And that leads to the very large increase in heat being generated. More recently, Lawrence Kazak and Bruce Spiegelman have shown that there can be this super stoichiometric feudal cycling in which creatine cycles between the phosphocreatine state, and that leads to the production of heat as well. Finally, within both brown, but especially white adipocytes, there can be triglyceride fatty acid cycling, where the triglycerides can become lipolyzed to fatty acids and then rebuilt, and this feudal cycling also generates heat. What remains to be determined is what proportion of thermogenesis happens from UCP1, from the creatine feudal cycling, from triglyceride fatty acid cycling, and other processes. It's important to remember, though, that adipose tissues, like every tissue in the body, are made up of a diversity of cell types. The single cell atlas shown here, which was published with lead author Margot Emont and led by Evan Rosen at Beth Israel Deaconess Medical Center and published in Nature just about 2 years ago, they showed a single cell atlas of human white adipose tissue. And there are many cell types. Besides adipocytes, there are stem cells, several types of immune cells, endothelial cells, and others, and they have distinct marker genes. So you can see here on the graph in the UMAP projection, these are the adipocytes in the adipose tissue, but then there are the smooth muscles, different white adipocytes over here, and the same thing is seen in the whole cell atlas as well. And these are the various marker genes that can define each of these various cell types. And each type can act locally, but also impact organs throughout the body. And that's something that I, as an endocrinologist, am particularly interested in, which is that the roles of adipose tissues are only beginning to be discovered. Shown here is a figure from a review article published by Phil Shear and colleagues, where they show that adipose tissue, for example, white adipose tissue, releases not just small molecule metabolites, but also non-coding RNAs, extracellular vesicles, the classic proteins like leptin and adiponectin, and also types of lipids. And shown here, for example, from a review written by Ron Kahn and colleagues, is that white adipocytes will release, for example, microRNAs that then influence the activity of muscle cells, pancreatic beta cells, liver cells, and then white adipocytes, white blood cells such as macrophages. So there is a lot of communication going on within the tissue and beyond. When it comes to brown adipose tissue, as an endocrinologist, we think of both the functional and the endocrine roles. So brown adipose tissue is a thermogenic organ. Cold exposure leads the sympathetic neurons to release norepinephrine, which then cause a breakdown of the endogenous lipids within the brown adipocytes. There's also a very large increase in blood flow that then leads to the delivery of glucose and free fatty acids into the brown adipocytes. These breakdown products then enter the mitochondrion, as we described before, and eventually heat can be generated. But besides the thermogenic function, there's the endocrine role. And already it has been shown that brown adipocytes themselves can release numerous types of factors. 12-13-dihome that influences brown adipose tissue and muscle fatty acid uptake. It also causes the release of myostatin, neuregulin-4, and other compounds. As a result, there are suggestions that even in humans, brown adipose tissue can act as an endocrine organ that has major impact on cardiometabolic disease. So how does one study a functional organ in humans, such as brown adipose tissue? Our laboratory has developed a research platform consisting of 3 pillars: non-invasive imaging, integrative physiology, and then therapeutics. Regarding the imaging, we use primarily PET-CT, which gives functional information. We're developing MRI and also ultrasound for different purposes. On the integrative physiology side, which is what the rest of this talk will be about essentially, we use both human and rodent models to look at bioenergetics, which is the generation of heat and lactic acid, bioinformatics approaches to both generate hypotheses and confirm them, and also gene editing and regulation studies to learn more about the roles of different proteins in physiology. And then the therapeutics is really the clinical trials. In our field, there's been successful application of mild cold exposure, and in particular, I'm going to focus on beta-3 adrenergic receptor agonists, in this case, mirabegron. This is the clinical trial that we published most recently regarding the assessment of mirabegron. It's called the Chronic Mirabegron Treatment, or CMT study. This is its code that you can find in clinicaltrials.gov. And in the first cohort of the study, our primary endpoint is the change in detectable brown adipose tissue metabolic activity, as seen by PET-CT, that is associated with daily dosing of mirabegron over a 4-week period. In this study, we recruited 14 women, otherwise healthy, and for 4 weeks, they took by mouth 100 milligrams of mirabegron each day. Note that this dosage of mirabegron is higher than what is approved to treat overactive bladder, which is the FDA-approved treatment. So please, this is not something that you should try at home. This is something that we're using only for our studies. And this research was led by Elena O'Mara and James Johnson, who had graduated from college and were in our lab as post-baccalaureate fellows, and now are physicians working in healthcare. And so in this study, we did a series of metabolic tests and imaging, both at the initiation, day 0 and day 1, and then at the completion of the 4 weeks of treatment on day 27 and day 28. Each person underwent an FSI-GTT, a frequently sampled intravenous glucose tolerance test, to look at both glucose tolerance, but also beta cell function in terms of making insulin. They underwent a liver scan called a FibroScan and CAP to look at liver fat and also liver fibrosis, an exercise tolerance test, DEXA to look at body composition. Each person entered the metabolic chamber we have at the NIH at 6 p.m. so that they would stay in a warm environment and not by mistake activate the brown adipose tissue. At 8 a.m. of the study day, they had their blood drawn. Mirabegron was then given an hour later, and then we drew blood for pharmacokinetic measurements. At the Tmax, the time we expected the maximum mirabegron exposure, we drew blood again, so there was a 5-hour separation to see how mirabegron worked. We then gave the people FDG, which is a radio tracer that allows us to see the brown fat. After an hour of going through the blood, the imaging via PET-CT was done, and then everybody got to have lunch. What did we find? This is a 28-year-old woman, and this is her PET image on day 1. The black is where the glucose is going into the cells. You can see a lot of black going into the brain at the top, and then some black here at the bottom going into the kidneys. And there's a little bit of FDG uptake corresponding to the black here in the supraclavicular region. After 28 days of taking mirabegron, there was a very large increase in the amount of detectable brown adipose tissue in the cervical, supraclavicular, and axillary regions, down the spine and the paravertebral zones, and also around the kidneys. This was a very successful intervention. And over the 14 people we studied, nearly everybody had an increase in brown fat measurements. Shown here are 3 different parameters that describe BAT activity. And you can see here on all 3 graphs, they're on a log scale. So there was a very large increase shown here in the green, which are the medians, in the BAT metabolic activity. It was greater than a doubling of brown fat volume, and the maximum BAT activity increased as well. So we satisfied our primary endpoint. But we went beyond that, and we showed that chronic mirabegron treatment, when looking at 8 a.m. on day 1 to 8 a.m. on day 28, there was an increase in the HDL, or good cholesterol, by 8%. There was also an increase in total GIP. GIP is an incretin, which is a hormone that increases insulin secretion in the setting of hyperglycemia. And that went up. Adiponectin, which is a hormone released by white and brown adipose tissue and causes increases in insulin sensitivity in skeletal muscle and liver at the very least. That led to that was increased by 35%. And then we found in the plasma an increase in total bile acids by nearly 50%. And we're particularly interested in the bile acids because, similar to adipose tissue, they're often thought of in terms of their function, and they're surfactants, these bile acids, and they help absorb dietary lipids, cholesterol, and medications. But at 1,000th the concentration in the plasma, these bile acids act as endocrine hormones that bind to nuclear receptors such as FXR, LXR, PXR, and cell surface receptors such as TGR5. And these receptors are found throughout the body. In the liver, bile acids can improve glucose, lipid, and lipoprotein metabolism. They can be cardioprotective in the heart. They can improve or at least modify the microbiome. In the brain, they are neuroprotective and may affect appetite suppression. There are changes in insulin sensitivity and skeletal muscle, increased BAT thermogenesis, and modulation of the immune system. From the frequently sampled intravenous glucose tolerance test, in collaboration with Richard Bergman, who developed this modeling system, we found that the glucose effectiveness increased by 34%. That's the capacity for glucose to mediate its own disposal. The insulin sensitivity index went up by 36%. And in terms of the pancreatic beta cell function, the acute insulin response to glucose, which is the production of insulin by the pancreas, increased by 37%. And the disposition index, which is a reflection of beta cell responsiveness, increased by 82%. Now, we have since used the plasma from the patients we've studied from different clinical trials in the past to then explore what activated brown adipose tissue may be doing. And this is done predominantly in collaboration with Yuhua Tseng at Joslin Diabetes Center and Harvard Medical School. And we found, for example, that the cold-induced lipokine coming from brown adipose tissue, 12-13-dihome, promotes fatty acid transport into brown adipose tissue and skeletal muscle, shown here in the diagram. In addition, 12-lipoxygenase, an enzyme in the brown adipocytes, leads to the release of 12-HEAP from brown fat and causes glucose uptake in other tissues. And finally, along the lines of this, we've shown that the brown adipose tissue-derived compound myosin-2, another bioactive lipid, contributes to cold-induced resolution of inflammation. So we are now seeing, using animal models and human clinical trials, the identification of several bioactive lipids that then impact metabolism. And so in summary, for the chronic mirabegron treatment studies, 4 weeks leads to the following. We see increases in BAT metabolic activity. We see increases in resting energy expenditure. I didn't show you this, but it was 6% above baseline. There were increases in HDL, total bile acids, and adiponectin. There was improvements in glucose uptake and beta cell function. And so therefore, targeting the beta-3 adrenergic receptor may be a new approach to treating obesity-related complications, such as type 2 diabetes, as well as components of the metabolic syndrome. But the big question is, how can the underlying mechanisms remain to be determined, given that BAT and WAT likely have important roles? And so this is where we turn to in vitro models. Shown here are a number of different types of models, and the challenge has been a difficulty in accessing and obtaining human BAT, and lack of suitable human brown adipocyte models, and those have hampered the understanding of the physiological role of human brown adipocytes. The few models that have been used so far include the multipotent adipose-derived stem cells, or HMADs, the multipotent mesenchymal precursor cells, pluripotent stem cells, primary culture derived from adult bat, and immortalized human bat cell lines. I'm going to show you one set of studies using primary human pre-adipocytes. These were developed in our laboratory by Dr. Cheryl Chetto. Shown here is tissue that was collected from a 16-year-old female who had died from advanced malignancy. And we collected the superficial neck fat and the supraclavicular brown fat. The gene expression profile shows that in gray, the supraclavicular fat has a number of the genes higher among those that are associated with brown adipocyte function and activity. UCP1, which I mentioned was the defining uncoupling protein within brown adipose tissue, in the supraclavicular tissue that we collected, the UCP1 expression was over 100-fold higher, and that was also reflected by the protein levels here. And so what Cheryl did is she isolated the stromal vascular fraction, and that's where one gets the fibroblast precursors. She then grew them in culture. And the reason why she used non-transformed cells for these initial studies is that they tend to resemble the original tissue characteristics in physiology, and their entopic or resident gene expression and protein function is much more similar. What Cheryl found is that when she knocked down the beta-3 adrenergic receptor called ADRB3, there was a reduction in a number of other genes that were expressed that are associated with bioenergetics. So there is a 90% reduction in UCP1, and then there was also a reduction in these other genes that are associated with mitochondrial function. With this knockdown in red, Cheryl showed that forskolin, which activates the adrenergic system, had very different effects. So without forskolin at all, there was a lower level of cyclic AMP, which is downstream of adrenergic signaling. So it would appear that just having a functional beta-3 receptor leads to some downstream signaling that's important for cellular function. Even more so when forskolin is added, shown here, when there is a knockdown of the beta-3 receptor, there is less cyclic AMP generated, and also in response to isoproterenol, which is a molecule that binds to the beta-1, beta-2, and beta-3 adrenergic receptors. Similarly, there was a reduction in glycerol release, showing that cyclic AMP is lower and also downstream lipolysis is reduced. And to get a better appreciation of the functionality of the cells, we used a Seahorse XFE96 analyzer to measure mitochondrial bioenergetics. And in this case, this is a quick review of mitochondrial respiration, and more can be obtained from their website. We have no connection to the company. This is just a tremendously helpful resource. We can measure basal respiration of the cells, add oligomycin to block the F1, F0 ATPase, and that by the drop shows us how much is ATP production, how much of the oxygen consumption is from proton leak. We then add FCCP, which is a uncoupler that allows protons to rush into the mitochondrial matrix. That then gives us the maximal respiration. We then add antimycin A and rotenone to block complex 3 and 1 respectively, and that can give us the spare capacity and also the non-mitochondrial respiration within a given cell. In this case here, you can see in this mitochondrial stress test, the black line represents the control, and the beta-3 adrenergic receptor knockdown is here in red. And you can see that clearly there's a higher level of basal respiration and also a higher level of maximal respiration in the control. In other words, knocking down the beta-3 receptor lowered several bioenergetic features within these cells. And so using a cellular model we were able to then learn a lot about human brown adipocyte function. So with this model, upon differentiation, these primary human brown adipocytes from the supraclavicular depot increase the expression of the adipogenic, beta-adrenergic, and thermogenic genes. We found functional beta-3 receptors, but then knocking it down, that beta-3 receptor led to lower basocyclic AMP, shown here by these lower arrows, also lower cyclic AMP-mediated downstream signaling and lipolysis, lower mitochondrial content and cellular respiration, and lower UCP1 expression and UCP1-mediated thermogenesis. Therefore, one can appreciate the tremendous power from these bioenergetic and other studies that come from the well-chosen in vitro model. The problem with this model is that it is non-renewable. And so therefore, we worked in collaboration with ATCC to generate immortalized clonal human pre-adipocytes, both white and brown. And a description of these cells has already been published in the journal Endocrinology several months ago. And this is the reference. This was also led by Cheryl Chetto, and again in collaboration with ATCC. And this shows the derivation of the cells. This time we used abdominal subcutaneous white adipose tissue for the whites, perirenal brown adipose tissue from the browns. The primary fibroblasts were isolated, immortalized with hTERT. Selection was done for puromycin. Clonal cells were then generated, and then morphology differentiation, UCP1 screening was done to provide the hTERT-immortalized human white adipocytes, CRL-4063, and the brown adipocytes, CRL-4062. And as opposed to many other reports you will see in the literature, ATCC spent a lot of effort to characterize these cells. First was a genetic characterization and a functional characterization. Shown here are the white adipocytes, shown on the bottom of the brown adipocytes. They do not express CD34, which is associated with a non-adipocyte cell type. Rather, there is CD73, CD90, and CD105. So all these are associated with mesenchymal cells, which is what we wanted. These are the karyotyping of the white and brown adipocytes after multiple passages. And they are not quite normal, but not very abnormal, which makes their behavior much more likely to be consistent what entopic brown adipocytes will do in a human. The immortalized pre-adipocytes were successfully lysated and grow with distinct doubling times. These are the BAT primary cells, and these are the immortalized cells on the top. These are the whites and the hTERT immortalized on the bottom. And shown here are the doubling times. So generally speaking, the white adipocytes, the white pre-adipocytes, grow in a higher rate of doubling than the brown adipocytes, but both are doubling within the amount of time that is useful for conducting experiments. For those of you who are into the details, I will show them to you here. The cells are grown in DMEM with 10% FBS, 1% penstrep, and HFGF. The white adipocytes have a distinct set of culturing conditions. There's an induction media, and that's for 7 days, including rosiglitazone. And then there's a second induction medium, number 2, which is from 18 to 23 days, leading to roughly 30 days from the time that differentiation is attempted to begin to when the cells are ready for experiments. The brown adipocytes have 3 steps. There's a pre-incubation step that involves different compounds, including human insulin, thyroid hormone T3, and bone morphogenetic protein 7. Then we have the induction media number 1 and induction media number 2, which are the same as the induction medias 1 and 2 that were used for the white adipocytes. Do these work? Yes, they do. You can see here at day 0, the white adipocytes look like pre-adipocytes look like fibroblasts. The brown pre-adipocytes look like fibroblasts on day 0. After 30 days, there's a large accumulation of lipid droplets, which you can appreciate in these light microscopy, but even better with oil red O staining. Oil red O binds to lipid droplets. You can see very little here on day 0 with the whites, very little with the brown pre-adipocytes on day 0. And then after full differentiation, the white adipocytes are taking up a lot of oil red O, and so too are the brown adipocytes. At the protein level, we see shown here are the white adipocytes on the left, the brown on the right, day 0, and then after 30 days of differentiation. There is expression of FABP4 and adiponectin in both the whites and the browns. UCP1 is seen only in the brown adipocytes as expected. PGC1 alpha, which is involved in mitochondrial biogenesis, is really predominantly in the brown adipocytes as expected. And here is the control expression of GAPDH. I'm showing you here a time course of the development of the white adipocytes from the pre-adipocyte stage. This is day 5 through day 30. You can get a sense of how long it takes for the lipid droplets to occur. As I showed you before, the oil red O is able to dye the cells quite substantially because of the lipid accumulation. And shown here is the gene expression. And you can see that generally speaking, the white adipocytes achieve high levels of adiponectin and FABP4 mRNA by roughly day 20. There is no UCP1 accumulation as one would expect. This is in contrast to the brown adipocytes. Certainly they look a lot like the white pre-adipocytes at day 0, and under the microscope they're similar by day 30. And shown here is the oil red O, but the difference is the gene expression. It may be a little bit earlier with adiponectin and FABP4, but shown here there is an enormous amount of UCP1 accumulation in these cells, as would be expected, and would predict a substantial amount of uncoupling thermogenesis in these cells, but not the others. For those who are interested, the thermogenic markers such as UCP1 and PPRGC1 alpha are much higher in the brown adipocytes compared to the white adipocytes. The white lineage markers are higher in the white adipocytes compared to the brown adipocytes. More mitochondrial markers in the brown compared to the white. Brown lineage markers much more in the brown compared to the white. And at the protein level, shown here is a Western looking at the different electron transport chain complex enzyme. This is CV, meaning complex 5, complex 3, complex 4, 2, 1. And you can see that there's very little protein in the white adipocytes prior to differentiation, much more afterward. Similar with the brown pre-adipocytes, very little expression, and then considerably more of all of these electron transport chain proteins after the full 30 days of differentiation. The mitochondria are also highly present within the brown adipocytes, and you can see the UCP1. So shown here is immunofluorescence imaging, mitotracker in red, UCP1 in white, lipid in green, DAPI for nuclei in blue, and then the merged image. And you can tell that UCP1 is not present in the gene, not in the protein, and only in the brown adipocytes. And that's appreciated also through the merged image. So the interim summary here is that we have generated immortalized clonal white and brown human pre-adipocytes from abdominal subcutaneous and perirenal depots, respectively. The differentiated cells express the genes and proteins standardly associated with each cell type. In doing so, we've established renewable and scalable model systems that have the molecular and structural machinery to investigate their cellular physiology. And so in the next set of experiments, we will now demonstrate the functionality of these cell types. So how do these new cells compare to other offerings from ATCC? And so we compared these new cells with adipose-derived mesenchymal stem cells, normal human PCS500-011. These are fine cells, and they are fibroblast-like and are best grown in mesenchymal stem cell basal media supplemented with mesenchymal stem cell growth kit low serum components with 2% FBS. These cells are multipotent, capable of differentiating into the adipogenic, osteogenic, meaning bone, and chondrogenic, meaning cartilage lineages. The cells are cryopreserved at the second passage, which is very early, to ensure the highest viability and plating efficiency. We took these cells and differentiated them using the standard cocktail that I showed you in the previous slide, but then the white and brown media that I described earlier to see what we could do with these cells. We see that there is a higher expression. This is day 0, and this is the 3 different media. There's a higher expression of FABP4 and adiponectin from 10 to 100-fold higher, or even almost 1,000-fold higher with both of these. So therefore, we were able to cause these cells to differentiate into whites. However, these mesenchymal stem cells cannot be induced to become brown. So here's HOXC8. This is a white marker. It's much higher in the white cells that I described previously. It's not in the brown cells, but these mesenchymal stem cells will demonstrate expression of them after differentiation. Similar with the TCF21 white marker. However, UCP1 is really only in the brown adipocytes, not in these mesenchymal cells, no matter what one of the three differentiation protocols is used, similar with other brown marker DIO2. So one must use these cell types in order to, the brown pre-adipocytes, in order to get bona fide brown adipocyte functionality. So I'm going to show you a diagram that will explain the functional studies I'm about to go through. So sympathetic neurons will release noradrenaline or norepinephrine. They bind to beta adrenergic receptors. That causes ATP to become cyclic AMP by adenylyl cyclase. Cyclic AMP activates protein kinase A, and protein kinase A then phosphorylates numerous proteins. You can see here that glucose can enter a cell through GLUT1 and GLUT4 transporters. And so forskolin activates adenylyl cyclase directly, bypasses the receptor. And that can then lead to an increase in phosphorylation of HSL, hormone-sensitive lipase, which leads to the production of fatty acids and glycerol that can then be consumed in the mitochondria to generate heat. It also leads to an increase in glucose uptake in these cells and an increase in free fatty acid uptake from the media or from the blood and then into the cell. And I show you here, once again, how the heat is generated with the fatty acids being consumed, the production of protons that then go to the intermembrane space, and then uncoupling can happen, or ATP generation. So upon differentiation, both the white and brown pre-adipocytes can then become mature adipocytes that synthesize the proteins necessary for lipolysis. Shown here are Western blots showing HSL, and you can see here that HSL is present in both the white and brown mature adipocytes. When forskolin is given at 10 micromolar, there's an increase in phosphorylation of HSL at 563 and at 660. And that shows that the proteins are responding to adrenergic stimulation. And when glycerol is measured as an indication of lipolysis, there's a very large increase in glycerol production in the white adipocytes and also the brown adipocytes, as one would predict. We'll now look at the bioenergetics, which is the uncoupling, and this is the feature of the cells that is of most interest usually for those studying brown adipocytes. The brown adipocytes are here in red, and the white adipocytes are here in black. This is after 30 days of differentiation, and you can see very clearly that the basal respiration of the brown adipocytes is much higher than the white, and that's demonstrated here in this graph. And these other 5 graphs show the quantification of the information shown here in the tracing. So basal respiration is higher in the brown adipocytes, higher proton leak, higher maximal respiration, higher spare respiratory capacity, and higher non-mitochondrial respiration. And these values are similar to the values I showed you of the primary human adipocytes that were studied previously, and comparable to the energy expenditure that one sees in mouse brown adipocytes that have been reported in the literature. What does forskolin do to cellular respiration in the brown and the white? Shown here is another oxygen consumption rate tracing. This is time on the x-axis. And you can see here the red is the brown adipocytes, black is the white adipocytes. And when forskolin is added, there's an increase in the oxygen consumption rate. The vehicle, there's no change. Similarly, there is an increase in oxygen consumption of the white adipocytes, not nearly as much, and more delayed as in the brown adipocytes. In this case, oligomycin is added, and that reduces the oxygen consumption rate, as would be predicted. And shown here are the bar graphs demonstrating that the brown adipocytes have a large increase in oxygen consumption rate that is higher than one sees in white adipocytes. Brown adipocytes also have a higher uncoupled respiration. And then a very detailed issue related to our field of brown and white adipocyte heat generation is what were the sources. As I showed you in a very early slide, UCP1 is just one of the hypothesized proteins that leads to uncoupling. There's also the fetal creatine cycling and others. Shown here is an experiment using these new cells that was published in this paper in endocrinology, where forskolin is used to then cause respiration to increase within the brown adipocytes. And then GDP blocks uncoupling protein 1, and that causes a decrease in oxygen consumption rate. And this is a dose response, with each color representing a increasing dose of GDP. The key lesson from here is that when one adds GDP, there is a reduction in the thermogenesis, but not completely. It's only 35% drop. And that is the same thing seen here with the brown adipocytes shown in red, and then GDP is added. And here are the ones, the whites in black, the white adipocytes shown in black, where GDP is added. And GDP has no effect on the white adipocytes, and that means that there is no impact on UCP1, which we know doesn't exist. So it's a nice control. Whereas there's a 35% decrease in the thermogenesis in the brown adipocytes, which means that by blocking UCP1-mediated fetal cycling thermogenesis at only 35 to 45%, it means that there is no effect on the white, just the brown. It means the majority of adrenergic thermogenesis in human brown adipocytes is not from UCP1. And for those who are studying uncoupling, this becomes very important, because UCP1 is regulated by certain pathways, while other fetal cycling is controlled by other signaling mechanisms. In collaboration with Jonathan Jacobs and his team at the ATCC, we did RNA-seq transcriptomics. And what that gives is genetic signatures of the different adipocyte states. Shown here is the 4-part Venn diagram. For example, the day 30 versus day 0 genes, day 30 versus day 0 white genes, and then the whites compared to the brown at day 0, whites compared to the brown at day 30. And so what this provides you is an opportunity to see, once you have the cells in culture, whether the gene expression profile is consistent with what we have shown already. And this is a dendrogram showing the relationship of expression of the different cell types. And just with unbiased sorting, one sees that UCP1 expression and the beta-3 adrenergic receptor expression are right next to each other, reinforcing the paper that we had published previously, showing that these two proteins are very intimately related to each other in terms of cellular physiology. And so interim summary 2, these human brown pre-adipocytes, once differentiated, have an overall increased cellular respiration, higher UCP1 expression, and mitochondrial content. Both cell types, the whites and the browns, have a functional lipolytic machinery and undergo cyclic AMP pKa activated lipolysis with similar phosphorylation of HSL, which is hormone-sensitive lipase, and glycerol release. RNA sequencing data are consistent with the PCR data and can be utilized to corroborate and likely identify new physiological relationships. And these data highlight new genes and pathways that are different in these two cell lines, opening up many possibilities of new research. Our final summary. First, adipose tissue is polychromatic. It can be white and brown and actually more colors, and each tissue depot contains multiple cell types. Many of the endocrine roles and their mediators remain to be determined. To help study the mechanism underlying the different functions, we generated and characterized a pair of immortalized, clonal, human brown, and white pre-adipocytes. The differentiated white and brown adipocytes phenocopied primary adipocytes in terms of adrenergic signaling, lipolysis, and thermogenesis. Transcriptomics via RNA-seq were consistent with the functional studies and established a molecular signature for each cell type. And so finally, these standardized cells are anticipated to become a common resource for future physiological, pharmacological, and genetic studies of human adipocytes. Thank you. I just want to make sure I give credit to the team, in particular, Cheryl Chetto, who led many of the in vitro studies that you see here today. Our current team consisting of Ashley Schmitz, Sahara Ali, Kelly Long, and Nguyen Nguyen. Alumni of the lab in yellow here, collaborators in DIOB, And in particular, I want to thank our collaborators within the ATCC, the cellular side led by Fang Tian, and the informatics side led by Jonathan Jacobs, many other collaborators with whom we've worked to establish the data that I showed you here. Thanks again.
All right, well, thank you for the excellent presentation, Dr. Cypess. In just a few moments, we will begin our Q&A session. To the audience, please use the chat function available through the webinar program to submit your questions. This session will be archived on the ATCC website at www.atcc.org. Oh, and I can see that our product manager that covers these products, Jonathan Sagel, has joined in. So he'll help out answering some of these questions. So our first question that's come in, it's very technical in nature. I will send Dr. Cypess's way. Are there special cell culture media requirements to expand and differentiate these pre-adipocytes?
Thank you, Dr. Shapiro. That's a great question. And the answer is yes, to the extent that there is a published protocol, which is the one that was described in the webinar on slide 27 and is in the manuscript that we published. And it's important to follow them because there are several media to use, and protocols that are a little bit similar but not exactly the same do not work.
Okay, that's very good to know. Very good to know. All right, Dr. Cypess, how can one access the RNA-seq data for the models?
The data are freely available. They're part of the manuscript's supplemental data, which can be accessed in reference 30 of the manuscript.
Okay, very good. And what was the rationale for developing these two lines from a donor with von Hippel-Lindau syndrome? And are there any specific application benefits from this donor background?
Right, so the choice of a patient with von Hippel-Lindau was not deliberate. Rather, the clinical trial protocol that we use to collect tissue from patient volunteers permits the collection of tissue from any patient undergoing surgery at the NIH Clinical Center. And as it turns out, there are many patients there with neuroendocrine syndromes. This happened to be a person who we work with. Now, von Hippel-Lindau syndrome, which has been associated with increased visceral adipose tissue, was not seen with the specific mutation in the patient from whom we collected tissue, V167Q. Therefore, there may be some specific genotypic and phenotypic behaviors that are distinct to these cells and not reflective of intact tissue or other in vitro models, but we do not know.
Okay, very good, very good. Now, Dr. Cypess, are there any application benefits from developing these two cell lines from the specific fat depots that were selected?
Yes, yes. So to our knowledge, the sources of the cells are distinct from nearly every other one that's been described in the literature, including the previously published model that we showed in JCI Insight from 2021, Chetto et al. Those other models used fat tissue collected from the upper body, particularly the supraclavicular region and the neck, because those are easy to access by surgeons. The advantage of the models that I just described, and we work with in collaboration with ATCC, is that the tissue is from the abdominal subcutaneous fat, which is not necessarily quite the same as the upper neck subcutaneous fat, and the perirenal adipose tissue from which we got the brown adipocytes. Those are also not typically accessible and behave differently. We do not know yet all of the differences, and that is something that one could compare our cells with others to find out what those are.
All right, very nice, very nice. Okay, this next question's coming in. We have quite a few that have come in, actually. Very interesting results. could you talk about the radio tracer that was used in the study?
The radio tracer was 18-fluorodeoxyglucose. It's a PET emitter, and it's used typically for examining patients who need to undergo surveillance of cancers because fluorodeoxyglucose behaves like glucose and goes very readily into highly metabolic tissues such as cancer cells, but also brain, kidney, heart, and, as I showed, brown adipose tissue when activated.
Nice, nice. Now, Dr. Cypess, what does the biotin do for the induction media?
Biotin is an essential vitamin, and it's important for amino acid and energy metabolism and fatty acid synthesis. So it's something that over many years people have determined that it's a useful component to add to culture media.
Okay, very good, very good. Now, I believe I'm going to send this next question over to Jonathan. Could you talk about what could be the other in vitro models for studying pancreatic disorder or obesity or diabetes?
Yeah, those are some great questions. Thanks for asking. So we do have some really exciting immortalized cells. We have a collection of over 70 different cell lines of many different tissues, including these recently added pre-adipocytes. There are, as mentioned, some other pre-adipocytes, some alternative tissue depots, which have been deposited. Could also be additional models. Specifically with pancreatic tissue, there is a collection of 4 items, which were posited from a research lab in University of Nebraska, where they immortalized cells from different regions of the pancreas. They've been extensively published on and can be found on our website.
All right, very good, very good. Now, this next question that's come in, I'll let Dr. Cypess start, although I think Jonathan might be able to expand upon the first part of the question. So the way the first part of the question goes, can you discuss the use of immortalized versus non-transformed primary cells for these purposes? And then the second part of the question, how would these different cell types respond differently to drug treatments in brown induction? Brown fat induction, I guess.
Would you like me to start on that one?
Yeah, sure.
Sure. Okay. So this is a fundamental question that cuts to the core of all kinds of questions about how you do in vitro studies. Immortalized cells have the advantage of being in larger number, and they can be maintained in a laboratory for many more months or years and be available, whereas primary cells are not. They will typically not have too many doubling times. The advantage, though, of a primary cell type is that it is much more similar to the behavior of the intact cells that would be seen within an organism. And that, for that reason, what we did in the particular manuscript is show how the immortalized clonal human brown and white pre-adipocytes after differentiation, how they behave in terms of the gene expression profiling, in terms of the lipolysis, but also thermogenesis. And they have many features in common with the primary adipocytes that we published previously. So at that level, we think they're a good model, but there are many ways, many not yet known, of how these cells could be different. Truth be known, every single model, and even every single subtype, if one has, for example, HEK cells, they're potentially going to behave differently from one laboratory to another one down the hall. So you always have to do a certain series of tests to confirm what the behavior is when you're studying a cell type. But in a general sense, the immortalized ones shown here have the key features one would want from brown and white adipocytes.
Nice, nice. And Jonathan, could you expand a little bit about, say, hTERT-immortalized cells versus primary cells, versus just continuous cells in general?
Yeah, that's a wonderful question. I think We have addressed this topic in a webinar that are available on demand if you want a full, detailed response. But generally speaking, the continuous cell lines, the classical cancers, the A549s, continuous, easy to handle, maintain a fairly stable phenotype, but they're not truly reflective of human physiology. The primary cells, the other end of the extreme, directly from primary tissues, normal healthy donors, and most are best suited to replicate human biology in a dish when studying them. However, they don't replicate, or they have limited replicability in vitro, and therefore don't provide a reproducible, stable model. We believe that the immortalization of primary cells, as described in this talk and many other models we have in the collection, offer the best of both worlds. They have that stable phenotype, maintain proliferation in vitro, but because they are sourced from primary cells and minimally transformed with the immortalization process, they maintain all the hallmarks of the biology, as shown by the detailed studies from Dr. Cypess here. Additionally, for many of the models we have in our collection, there are publications, application data also demonstrating that the functionality of the immortalized cells recapitulates the hallmarks we anticipate to see in the tissue, the primary cells derived from those tissues.
All right, very nice, very, very complete explanation. Thank you. All right, back to Dr. Cypess. Has your group at all explored the insulin signaling cascade and insulin stimulating glucose uptake in the two cell types that we described today?
Partly, yes. We have begun these studies. So we've shown, it wasn't in the manuscript, but we already have preliminary data in our laboratory that adding insulin at 100 nanomolar leads to an increase in AKT phosphorylation within the white adipocytes. So that we've already begun to show. We have not yet done the experiments to look at insulin-stimulated glucose uptake. It's something we're interested in doing, and others may be already doing that as well.
All right, great, great. And have you considered expanding human patient studies? So in each of the parameters that you've shown, there were at least 2 with a reduction in the measured parameters. Have you traced them, and do they belong to the same people? Any idea why?
That's a great challenge to any time you do a clinical trial. Is not everybody responds as hypothesized, and why are they different? And sometimes we can get a good answer as to why. For example, we know that their body processes fatty tissue differently, and that you can get sometimes from their patient history. In most cases, we don't know why there's a different behavior. We have permission from many of our clinical trial volunteers to do whole genome and whole exome sequencing, and we've banked that for all the volunteers I've prescribed. We haven't analyzed it yet, but that's something that we could do. And hopefully, for many of the more intriguing behaviors that are not as predicted, we'll get some answers.
All right, nice, nice. Now, this next question goes back to the first part of your talk. Have you tested lower concentrations of mirabegron in human adipocyte browning? And what are your thoughts on mirabegron as a potential anti-obesity drug? That last bit, actually, you may not be able to answer on this particular
I have to give, yes, and so the most important thing to state is that the dose of mirabegron that I showed you, either 200 milligrams in men or 100 milligrams in women, is above the dosage permitted by the FDA for treatment. And so everything I showed you should not be tried with patients until safety would be confirmed. So that's the most important thing. The second thing is that, yes, we did try acutely other doses of mirabegron. This is a paper published in 2018. I, Aaron Cypess, am the senior author. It's in Diabetes, the journal, and Baskin, B-A-S-K-I-N, is the first author of that paper. And we looked at people who had acute responses to placebo, 50 milligrams of mirabegron, which is the highest approved dose for overactive bladder, and then 200 milligrams of mirabegron. what we found is that 50 milligrams had a statistically significant increase in activating brown adipose tissue, but nowhere near the same amount as 200 milligrams in men. Therefore, we think, and we believe the reason why there's such a difference is that at the higher doses of mirabegron, there's much more plasma exposure, and when it comes to the beta-3 receptor, there's not nearly the same level of expression in human white and brown adipocytes compared to rodents. And the tissue with the highest expression of the beta-3 receptor is urinary bladder. So in the end, we personally don't know what chronic treatment with a lower dose of mirabegron would do. It might lead to some browning. Could mirabegron be used as a potential anti-obesity drug? Potentially, yes. And much more research needs to be done to find a way that is both effective and safe.
Okay, very good, very good. Now, this next question that's come in, actually, first, we're over 1 o'clock. Dr. Cypess, do you have some time to answer just a couple more questions before we close?
Yes, I'm available. Yes, happy to.
Excellent, excellent. Okay, so this next question is about scalability of the cell lines. Would it be possible to use these cell lines, the hTERT-immortalized brown and white pre-adipocytes, for developing cell-based high-throughput screening assays to identify therapeutic small molecules? And if so, among the assays that you showed, which readouts do you suggest to use in a high-throughput setting?
We have considered this application. It was one of the principal reasons why we developed these cell lines, because they can go through many population doublings and retain many of their basic features after differentiation. We have not done any high throughput screening assays to develop therapeutic small molecules. The specific readout would depend on what one wants to see, whether it's the production of certain adipokines, for example, the bioactive lipids 12-13-dihome or 12-HEAP, or is it the increase in cyclic AMP, or is it an increase in oxygen consumption rate, and many others. So yes, we think these are very good models for high throughput screening, and what to look for depends on your goals.
Okay, great, great. Now, this next question looks like someone who is very interested in lipid biochemistry. Have you looked at the sphingolipid levels of the white and brown cell lines? Did you see any abnormalities?
That is a great idea. We have not yet done those experiments.
All right. And sort of a follow-up almost for lipolysis, talking about lipids. Do you prefer to measure glycerol instead of free fatty acids?
Yeah, we measure glycerol. You have to add a certain number of factors to the media to make sure that you don't get a false signal. But yes, we measure glycerol, and the specific details of the methodology can be found in the manuscript in endocrinology.
Okay, great. And let's see. Maybe you could sort of re-explain or sum up how you could use these cells for studying obesity, diabetes, and other chronic illnesses as our last question.
Well, with regard to the questions of obesity, it's predominantly issues related to the white adipocytes and white adipose tissue, because that's the principal site of energy storage. One has to remember that at one level, obesity starts with the brain and the perception of satiety, and then the brain will send signals out and identify hunger, et cetera. So when one gets to the the adipose tissue, it's really the white adipose tissue and the white adipocytes. So for example, we know that if white adipocytes are capable of expanding more, then that is a more safe response, because that prevents spillover of fatty acids and other lipid particles back into the blood, and that leads to ectopic lipid storage. Therefore, if one used these white pre-adipocyte cell lines and differentiated them, one could identify what enables a white adipocyte to store more triglycerides. You could also ask the question, which signal transduction pathways are triggered in the setting of storing too much triglyceride that then leads to the release of inflammatory mediators into the blood. There are many more things that one could do, for example, bile acid metabolism and looking at lipoproteins. There's so many things that obesity leads to as a consequence of too much triglyceride storage and other parts of dysfunction of the white adipocytes. One could study that there. With brown adipocytes, we are particularly excited at looking at them in terms of their ability to consume fuels such as glucose and fatty acids, and also the elaboration of hormones, we call them brown adipokines or batokines, that may be involved in regulating metabolism within the body. And there's already been a series of papers from multiple groups showing that both white adipocytes and brown adipocytes are communicating with other organs such as the liver, skeletal muscle, possibly even the brain, to give an indication of what the energy status is and what needs to be done.
All right. Great. Well, at this time, we will conclude our Q&A session. I'd like to thank Dr. Cypess for the excellent presentation, and thank you everyone for attending this webinar. The video and PowerPoint presentation will be posted on the ATCC website within 24 hours. For more information and to order the hTERT-immortalized pre-adipocytes, you can navigate to www.atcc.org/hTERT. Also, to read Dr. Cypess' paper featuring this data, you can follow the link in the resources or navigate through PubMed or Endocrinology's website. Thank you again, everyone, and have a great day.